Today is the first in our summer seminar series called Plant Phenomics Fridays. Some of you know about Plant Phenomics Fridays and you've done this before. Some of you may not know about it because we haven't done it for a few years. But the idea of Plant Phenomics Fridays is this. Sometimes in the summertime, you've got a little bit of free time. Fridays are the day that you're gearing up for the weekend, and maybe you want to take a little bit of time to just think and engage. And that's what we're doing in the space of phenomics for plants. The seminar series that we've set up for the month of August is every Friday, August 6th, 13th, 20th, and 27th. The screen that I hope you see right now is phenomics.iastate.edu. And on that page, you can see who it is that we have speaking, as well as the projects that are supporting Plant Phenomics Fridays this year. And let me scroll down to the bottom and show you that. We have two projects. The first is called HIPS, High Intensity Phenotyping Sites, a multi-scale, multimodal sensing and sense-making cyber ecosystem for genomes to fields. That's who is bringing all of us together. We also, so that's funded by USDA NIFA. We also have a newly funded project that maybe Baskar will just give a second or two talking about what IRA is. It's new. It's a team of us across a number of different universities. It's the AI Institute for Resilient Agriculture, funded by USDA and NSF together. So this week, we have Baskar and Erin Sparks speaking in the opposite order. So Erin's going to go first. And the way we're going to organize this, we're going to talk about it, is that in just a few minutes, Erin will get started and have a 20-minute seminar. If there's time for questions, we'll take a few. And then about halfway through at 12:25 Central, Baskar will take up the mic and tell you about what he's working on. And then we have 15 minutes at the end for discussion. So I'm very hopeful that in addition to asking some questions, perhaps at the end of their seminar, if you have thoughts that pop into your head, things that you want to talk about to this group of people that signed up and joined us, please think about holding some questions and ideas until the discussion so that we have a rich and meaningful discussion among this group of people. So our first speaker is Erin Sparks. She's in the Department of Plant and Soil Sciences. This is at Delaware. She's an assistant professor, and she's at the Delaware Biotechnology Institute and the University of Delaware. Erin got her PhD in cell and developmental biology after her BS in biomedical engineering. And she works, I would say, on something that I have a lot of fun seeing in the field, brace roots. And hopefully, and I can't imagine that it won't be the case, she'll tell us a bit about brace roots and other items on the topic of phenotyping plant mechanics.
First, I want to say a big thank you to Carolyn for the invitation to be here. And thanks for everyone showing up. I'm excited that we're able to resume these Plant Phenomics Fridays. I know I enjoyed them when they happened a couple of years ago. And so I'm really excited to see them start again. So I'm going to talk mostly about tools today, a little bit about brace roots. And what we're interested in is this idea of phenotyping plant mechanics. So really at the basis of this is understanding and preventing lodging. So if you're familiar with crops, lodging is basically the failure of plants to stay upright. So what you're looking at on the screen right now is a picture from Delaware last summer. We had a tropical storm roll through and basically blew over a lot of our cornfields. I know in Iowa, the derecho came through and caused a lot of lodging, a lot of crop loss as well. So we really want to dissect and understand the biology of this and the biomechanics of lodging. And we mostly work in corn and somewhat we're transitioning into sorghum as well. But if you go and you try to find metrics on lodging from an agronomic perspective, it's often reported as a single metric of 20% lodging, 10% lodging, but it doesn't distinguish between the different types of mechanical failure, which become important for us when we're considering how the plant fails mechanically. So lodging broadly can occur to two different classes. One is through root failure and the other is through stalk failure. So root failure is basically a failure of root anchorage. So the roots either break in the soil, they pull out of the soil, and you see the entire plant will fall over. In contrast, stalk lodging is a breaking or buckling of the stalk. And now I'm going to talk first about stalk failure a little bit. In maize, there's two distinctions of stalk failure. One is what's called green snap. Green snap occurs approximately mid-season, and it is breaking at the node of the maize plant. It often happens early in the morning when the turgor pressure in the plant is highest, and has been recorded to cause between 15 and 53% yield losses. In contrast, late stage maize stalk lodging occurs through a buckling mechanism. So in the internode, you can see here, this is the stalk buckling, and this is what is generally referred to as stalk lodging, and is estimated to cause between 5 and 20% yield losses. And I should take a moment to acknowledge the reason that these illustrations are so beautiful is a former PhD student in the lab, Lindsay Erndwine, who has just an amazing talent as a scientific illustrator. In contrast to those very clean, this is the percentage when the stalk breaks or snaps or buckles, root lodging effect will really vary based on the plant stage. So I mentioned that root lodging occurs when roots are either pulled out of the ground through uprooting or breakage of those roots. And there's this study from 1988, which is one of our kind of cornerstone studies that we look at. And basically what they did is at different plant stages, they pushed plants over and they asked what was the impact on yield. And basically what you can see is, if you're not familiar, V stands for vegetative. So a V10 plant would have a vegetative leaf 10 stage, and you can see these yield losses become exponentially greater as the older the plant is when it falls over. And you can see that here, a lot of that has to do with this phenomenon that we call goosenecking, or the reorientation of the stalk. So the older the plant is, the farther up the stalk this reorientation happens, prevents combine mechanical harvesting, which is one of the major sources of yield loss from root lodging. Is basically farmers don't want to go out and manually harvest their crops. So this is all well and good, but lodging itself is incredibly difficult to study because it requires a fortuitous event, or unfortunate event if you're a grower, like a severe storm, to come through and basically blow over your plants. And while these are becoming more and more frequent, it makes designing controlled studies practically impossible. So instead, there's a lot of effort to design proxy measurements of plant biomechanics that tell us about lodging resistance and how a plant may resist these external forces. So these proxy measures generally rely on the principles of biomechanics. And so to give you just a very introductory overview of how biomechanics works, you apply a displacement. You can apply this displacement in different ways. You've heard of probably tension and compression or bending. And then you're measuring the amount of force it took to displace that object the same distance. So a stiffer plant will have a steeper slope versus a more flexible plant will have a shallower slope here. So the first phase of plant materials function as an elastic or viscoelastic. So this is the equivalent of a rubber band. So when you stretch or change the material, if you release it within this elastic region, it will go back to its original conformation, and you haven't produced any permanent changes. If you continue to displace past this elastic region, you now start to see this failure mechanism. So we're having plastic deformation here, which is basically permanent deformation. And so we can get several metrics out of here, including the ultimate load, meaning the maximum force that the object can take, and then the break load, which is the force at which obviously your object breaks. We are primarily interested in things in this elastic range. So our goal is non-destructive measurements that can be used repeatedly in different contexts. So I want to talk a little bit about some of the devices that exist and how we're improving upon them. When we first talk about stalk biomechanics, again, I'm just talking in the context of large grains here, primarily maize. There are different considerations if you're working on small grains based on the mechanics of the plant itself. But so this is one device that has been reported in the literature, and basically you use a belt to strap on this force sensor to the plant, and you pull in all the different directions and you record the maximum force. The other device that we have used extensively for several years is called the Darling, I always forget these acronyms, device for assessing resistance to lodging in grains. And this can be destructive or non-destructive, and we'll talk a little bit more about how this works in the next slide. But again, the idea that you're pushing or pulling on a stalk and you're measuring forces. So this is Lindsay Erndwine, the former PhD student that I mentioned that does all these illustrations. And here she is demonstrating one of these Darling devices on a very senesced, very dead maize plant here. And you can see that, she's using this handle to kind of push forward on the plant. If we break down the kind of key components of this device, there is a load cell that's measuring the forces. And then at the base, there's a potentiometer that's measuring rotation. So we're getting out, instead of a force displacement, a force rotation curve. And we're, again, working in this elastic region. So we're just repeating these measures. We've confirmed that we're not permanently inducing deformations into the plant. So we've used this pretty extensively to look at how brace roots contribute to this metric by measuring and then cutting off brace roots and measuring again and looking at how the forces change. But in our maybe 3 years of using this device, we've identified some great things about it, and we've identified some things that we wanted to improve upon. So some of the pros of this device are that it's portable. So it's powered. One of our biggest challenges, and I'm sure many of you can relate to this, is to power our devices in the field. And so there's a battery pack on the side of this that will power these devices basically indefinitely. They're pretty inexpensive. It's just 80/20 aluminum framed and Raspberry Pi. There's a visual display, which is absolutely key. You can actually assess your data in real time. And I should note that these devices were built by Doug Cook, who is a professor at Brigham Young University. And so all this work was done in collaboration with Doug. I say these are medium throughput because you can go as fast as the person testing, but we'll talk in a second about why that's not always the greatest thing. And then the big plus is that the measurements from this device have been associated with stalk lodging and stalk lodging resistance. So all of these things that we're measuring, the key is to really tie them back to those lodging phenomenon that are unpredictable and hard to control. But some of the drawbacks that we've encountered are user to user variation. So I talked about the throughput. And the faster a person goes, the worse the quality of data. So the device is collecting data at a specific rate. And so the slower you test, the better your data, you have higher density data. And so we can plot it out and we can see the user variation in this data, which is not ideal, right? We want it to be mostly uniform from user to user. The other thing we've noticed is that because it's 80/20, there's some flex in the instrument, particularly at this arm out to the load cell. We'll see a little bit of twisting if the plant is really stiff. The other challenge has been the rotation measurement. So I mentioned that there's a potentiometer at the base, but what happens is that potentiometer is sitting at the hinge, and the more times that hinge opens and closes as we test, the looser the potentiometer comes, and it basically gets off scale. So we ended up not being able to use our potentiometer measurements, and instead we used an accelerometer measurement, which is up in the head unit here. But an accelerometer is even more sensitive to the speed of testing than the potentiometer. So there were some trade-offs in how the rotation was measured that we wanted to address as well. So this year, this is our new device. We haven't named it yet, but it's on a mobile cart. Some of the things that we've done is we've built a linear actuator. You can tell this is now completely hands off. So we're trying to remove that user to user variation. The load cell is on the front of this linear actuator. This is just a lead screw being driven by a stepper motor. And if I can catch it right at the end, there we go. You can kind of see here this data of the pushing and measuring the extension of this arm and then the forces that are being measured. So again, first time it went out this year, we've had some fun gremlins with it. Pros are that it's portable, kind of. It's on a cart, it has wheels, but this cart is extremely heavy. It's basically being powered off a car battery right now through an inverter. It's not the most portable of devices, and that's kind of one of our next areas to tackle, is to try to streamline this a bit more. It remains inexpensive. This is Arduino powered, again, a lead screw, a stepper motor. These are pretty inexpensive components. We have a visual display. I gave it a small check mark because in this case, it's a laptop again. And one of the big challenges we have with taking laptops in the field is that laptops are often black and they will overheat pretty quickly in the field. And again, medium throughput. Right now, our throughput is set based on the amount of time that it takes to extend that lead screw, and I'll keep playing that, and come back. But the keys for us is that we've now reduced that user variation, because you click start and you can basically move away, and the extension is going to be exactly the same every time. By not offsetting the arm, we've removed any flex in the instrument. And again, we don't need rotation measurements because we can extract displacement based on the extension of this arm. So we recently got an NSF grant to look at lifespan biomechanics of maize plants using this device. And so we've just started to do that in the field. We have some fun gremlins in this where it will randomly power itself depending on how we have things wired. So it has some room to grow here in terms of understanding and optimizing this device for broader use. But then the last 5 minutes, I want to talk about the other device that we've been building to look at anchorage. So at the beginning, Carolyn said that, my lab works on brace roots. That's what we're interested in. And so we really want to understand how brace roots help to anchor the plant in the soil. And if you go back and you look at previous methods to assess root anchorage, they've involved either pulling plants out of the soil and measuring the amount of force it took to pull the plant out of the soil, pushing plants over and measuring forces, or this kind of complicated pulley system to try to induce rotation at the base instead of these pulling and pushing forces. Major challenges have been encountered with these. These have been around since the early 50s, these types of approaches. They're all cumbersome. They're all destructive. They rely on actually pulling that plant out of the soil. And depending on the paper, they've had a positive association with lodging resistance, no association with lodging resistance, negative association with lodging resistance. And so they've been kind of all over the place. So we set out to design a new device that would be non-destructive and measure the torsional stiffness of the root system, which is basically the ability to resist rotation within the soil. So this device pictured here is called the Smurf, sorghum and maize under rotational force. And I'll show you a video in the next slide of how it looks, but basically we strap it onto the maize plant. We have 2 points of attachment because we don't want to measure the bending of the stalk. And so we've made the stalk rigid by having these 2 points of attachment. The foot here on the bottom will extend and actually apply a rotation at the base. So right off the bat, cumbersome nature has been reduced. This is a tiny device. It's all 3D printed, incredibly portable. We have a battery pack on the back here, and it communicates with a laptop through a radio chip. Non-destructive, these are measuring within that elastic range that I talked about. And then in a couple slides, I'll get back to this idea of, is there a relationship to lodging resistance? But here I wanted to show you a video. So the Smurf here is off to the right, and this is the base of the plant. We see our beautiful brace roots, and you can see as the Smurf engages, you can see these roots kind of bending, and it's most obvious when it relaxes at the end. And you can see how far these roots have bent, right? As they change, as the torque is basically applied. But does it have anything to do with lodging resistance? And so we are super grateful to Nathan Springer and Candy Hirsch. They put out a paper with Sarah Tirado as the first author, where they had assessed root lodging in the genomes to fields hybrids. And they were, in this paper from this year, they were assessing basically root lodging and recovery by changes in plant height by UAV. And so what they provided us with was seeds for 11 of these hybrids that we planted in our Newark, Delaware location last year. We have them out again this year. And conveniently or inconveniently, we had this tropical storm that came through last year and caused root lodging. So we were able to verify their findings that there was a variety of root lodging susceptibility within these hybrids. And so this is just these hybrids and percent lodging. We divided them into 3 classes: the low, which basically less than 25% of the plants lodged; high, which is technically medium, where greater than 50% lodged; and then the ones where they were 100% on the ground, we couldn't do anything with. We've kind of ignored those for the moment. And what we did is we took these Smurf measurements, we extracted the torsional stiffness, and what we found was that the plants, the hybrids with higher root lodging, had higher torsional stiffness. So they were less resistant to deformation than the low root lodging hybrids. So what this tells us is that the ability of the root system to flex within the soil is actually a critical component to be able to resist root lodging. And if you talk, we have collaborators in civil engineering who say, of course, this is a fundamental concept of buildings, that you want a base that's able to have some flexibility and not be a rigid base. So we're happy to see that, at least in our first pass, we're still collecting more data here, that we have now a very clean relationship between the root torsional stiffness and whether they're resistant or susceptible to lodging. So ultimately, our goal with this and building these tools and building these devices is we want to be able to link phenotype to function. So I'm not going to talk about it, but we also have this ground-based phenotyping robot that we call the Brobot. The brace root robot has a camera on the front here where we're taking images of the brace roots twice a week, the entire growing season, so we can understand how these phenotypes of the brace roots are linked with these anchorage measurements that we're taking with our Smurf. And so this is, the overview of my research program is understanding brace root biology. And so I'm talking particularly about the tools we've built to assess brace root function and the biomechanics. We also have some projects understanding their development and how the environment is impacting these as well. So with that, I will take a moment to thank our funding sources and all of the wonderful current and former lab members and collaborators. This is a big collaborative effort that have allowed this to happen, and I am spot on time. I don't have time for questions. We'll do them at the end.
Thank you, Erin. That was great. I have one tiny comment for you as we turn things around and get Baskar up here.
Yes.
I thought Smurfs were blue, not green.
You will be happy to know there's a baby blue one in the basement right now.
Fantastic. Dr. Baskar Ganapathysubramanian is a professor in the Department of Mechanical Engineering at Iowa State University. He has a bachelor's degree in mechanical engineering from the Indian Institute of Technology, an MS in mechanical and aerospace engineering from Cornell, as well as a PhD in the same area from Cornell. Baskar has this new project I'm hopeful he'll tell you a little bit about, because it's part of what our funding is for Plant Phenomics Fridays today. His seminar is entitled Reducing Annotation Workload for Plant Phenotyping. With that, here's Baskar.
Thank you, Caroline, for the invitation to speak. And I'll try to follow up on the excellent talk that Erin just gave us. So I'm going to talk about how we can use machine learning to extract out plant phenotypes, especially in the context of large field experiments. And as I will allude to, one of the things that that's a pain point, if you will, in using machine learning tools is the need to create a large training data set, right? And that could take weeks, if not months, of painstaking effort from staff and undergrads and grads. And so the idea in the past couple of years that my group and other groups across the globe have been thinking about is, are there strategies of reducing this annotation workload for plant phenotypes? But before that, a little bit of introduction to what my group does. I run a computational sustainability group. So we are essentially an applied mathematics, scientific computing, machine learning group with broad interest in sustainability applications. So broadly defined, we work on various aspects of energy, health, water, and in the context of this talk, food. So what do? We seek to collaborate with various domain scientists, and we want to develop and deploy computational tools for assessing, understanding, controlling, and perhaps designing complex systems. Right. So a lot of our work is funded by a variety of federal agencies as well as the state of Iowa, and I want to acknowledge their support here. So, like Caroline had alluded to, the new project that we are excited about, it's a group of 8 universities and commodity groups across 33 different researchers. Is the recently announced USDA NIFA AI Institute. AI institutes are the flagship AI centers, if you will, across the US, and one of them has been awarded to as a lead at Iowa State on AI for resilient agriculture. So I encourage folks to look at that website over there. The basic idea here is to develop over the next 5 years enabling AI technologies that will provide a foundational advance towards breeding as well as production. And a central idea here is how do we extract out enough traits, physiological, environmental, management, and link that with what we call a digital twin at the plant scale as well as the field scale. And so having a virtual digital twin essentially allows us to do a lot of things on a computer at a significant fraction of the time, money, and effort required, right? So you can evaluate different what-if scenarios under climate change. We can test out various hypotheses, and then we can identify or subselect a small set of actual experiments to perform in the field, right? So the key theme here is foundational AI and machine learning advances, but anchored with questions in agriculture. And so with that preface, I want to jump right into the specific subtopic I want to talk about today. That is AI ML, more broadly defined, enabled plant phenotyping. Right. So over the past decade or so, it has become clear that machine learning can be a game changer to helping us understand and more carefully tease out, say, G by E and G by E by M relationships. Right? So with the advances in remote sensing, high throughput sensing, the various types of sensor technologies, not just visual, but even VOC, hyperspectral at multiple scales, and like Erin talked about, more mechanical testing devices, we are able to extract a huge amount of information, especially from large field experiments. So the bottleneck is no longer the collection of data, though several of my plant science colleagues will agree to disagree on that, but it's rather creating enough well annotated data to train a machine learning model that can then extract out more and more and more information. Right. So to give you a perspective, right? So if you want to measure, you can take an image of a plant, but if you want to extract out, say, detect whether flowering is happening or not, you have to train a machine learning model to actually figure out when flowering is happening. Right? And for that, someone has to actually go and annotate When you say machine learning model, you have to annotate, maybe take 10,000 images, figure out where in those images there's flowering happening, and then teach a machine learning model essentially to say, if you see this kind of pattern, that means flowering has happened, right? So train a machine learning model, then you need to have annotated data. Right? And creating this annotated data is where the current bottleneck is. So I'm going to explain that with a research vignette here. So this is a most of the work that we do in my group is heavily collaborative. This particular project is with Pat Schnabel's group, Dan Nettleton, Yumo Chi's group at Iowa State, as well as James Schnabel's group at UNL. I have the pleasure of presenting a lot of this work. Most of the heavy lifting and the work is done by the guys, students, and the staff right there, they're in bold. And I thank them for their tireless efforts on this. So the basic experiment here is you have a stationary camera, about 450 or so stationary cameras that take images of one row of a specific genotype of maize. So this is both from ex vivo plants as well as I think the parts of a diversity panel. So there are about these images have been taken across the complete growing season. And you have about for the past 5 or 6 years, so you have about 4.5 million images across 2,200 cameras. So what is the question that we want to look at, right? So here, because we have time-lapse photographs of a plant growing, we can actually extract out time-dependent development traits, right? So easiest thing to think about is how does the height change as a function of time? Second thing is what is the leaf appearance rate, for instance, right? So what is the number of leaves that you can see at every day, and track that as a function of time. This can have very interesting implications both from the management as well as from understanding and revealing the genetics of these. So the first step towards extracting, say, the height of a plant or the number of leaves that you see in the plant is what we call semantic segmentation. Right? Segmentation essentially is saying you're given an image, you identify the key feature of interest. Right? In this case, in the figure on the right, you see I have this scene, and I want to identify where the plants are. Right? And someone has actually gone in and painfully marked all the plants with a blue pen. Right? So this has to be done very carefully because we want to train a machine learning model. Any mistakes you make, it's going to learn those mistakes too. Right? So the basic idea of semantic segmentation is shown in the figure on the left there. You go through pixel by pixel by pixel on an image, and you classify every pixel. Right? Is it a pixel of a background? Is it a cat, or is it a dog, for instance? Right? So that's where a lot of the machine learning advances have been made, differentiating between cats and dogs. Right? So in our context, we want to at least the simplest problem is you want to distinguish between does this pixel belong to a plant, or does it not a plant? So background, or is it the foreground or the plant? So now if we are able to do this, this can actually fit into a broader workflow that I will not talk about, but I briefly allude to here. You take an image, you take a time series data of the images across the growing season. You're able to segment out that image. You're able to extract out various traits: height, number of leaves, angle of the leaves, and like Erin talked about, the displacement of the stalk as a function of wind speed, and so on. Right? So you're able to extract out a diverse variety of traits from this. But the first step, like this whole talk alludes to, is that you need to annotate. You need to create enough data of the segmented images to train a machine learning model. And we tried this, right? We tried having both undergrads, grads from my group. They gave up very, very quickly. And then undergrads and grads from the plant science domain, they're a lot more enthusiastic about this. They persevered for 200 or 300 images, and then it just became, it's not just worth their time, right? So it's very laborious, it's time consuming, it's expensive. And then as one of the papers that several of us have published a couple of years ago, it also shows that this can be error prone depending on who does this, right? So if you're very vested in this problem, then you end up doing a better job than just having a crowdsourced random person try to do this. So what is our strategy here, right? So one of the things that I want to push to the plants phenomics community, the phenotyping community, is this idea of the new idea that has been in the machine learning field for the past couple of years called domain adaptation, right? So instead of actually creating painfully an annotated data set of the real experiment that you want to look at, why don't we artificially create a synthetic data set that looks very similar to the real data set and train our machine learning model on this artificial data set and then deploy it on the real data set, right? And this is something that has been used with great success in autonomous vehicles, right? So if you're training a self-driving car, you want to segment out and classify the scene that a car is seeing, the camera of the car is seeing, into that's a pedestrian, that's not a pedestrian, that's a signal, that's a road, that's a walkway, and so on. But doing this is again painful. And so what people have tried to use is they have tried to use realistic video games like GTA, right? That's what you see on the left, right? So they use scenes from a video game that is hyper realistic, and then they annotate because it's generatively created, right? So the video game knows exactly what each pixel is because it's actually making those things there. You have this large data set, and you can use this data set to actually create to train autonomous driving algorithms. We use the same idea here. We take ideas from that field and apply it to the plant phenotyping field, where we actually use data of plants imaged in indoor conditions where there's good lighting and you have individual plants, and you can easily segment out those plants without human intervention. And then we somehow try to place those segmented plants in a realistic background, right? So how do we do that? So the idea is Kaushik and Zaki and a few other people from Pat's group, we actually went out to the field, removed out about 117 unique plants, and then we took them indoors. We imaged them here this was a couple of years ago, in a red background. Now we use a blue background to have maximum contrast. And then we are able to actually segment out the images, right? So the figure on the far left is an actual image. The figure in the middle there is a segmented image. That is, we remove out all the background and only show where the plant is. And then additionally, we pick about 250 random empty backgrounds of an actual field experiment. And so the idea is that then what we do is we do two things. Step number one, we do what is called a color and style transfer. That is, we take the image that we see on the top left there is a segmented image. We take a random background, and then we use the color and illumination of what we see in that random background and change the color of slight color of the plant, right? So what that does is it makes it look like the plant is not in a controlled lighting. The plant is actually in illumination that can be actually seen outward. So now we have 200 different background images under different illumination conditions, when there's light, when it's overcast, when it's bright. And so you have this image, this plant that's measured indoor, but now it looks like it has been taken outside and it's been imaged. Once we do that, then what we do is we actually take a set of 5 or 6 randomly we select it from these plants that are measured indoor, and then we embed that into the scene from outside. Okay, that's what you see here. So you see these are not super realistic, but I guess they will do for training. The question is, will they be enough to train a machine learning algorithm, right? So now for each of these plants that you see, I should probably use this, each of these plants that the scene that you see here, you take a random background image, you randomly pick 6 plants that you have imaged indoor, and you essentially place them right here on the scene. The advantage of doing this is now for each of these images, without any human intervention, we have the semantically segmented plants. So to give you a perspective, doing this for 200 images took us about 3 days. When I say us, I mean the graduate students and the undergrads. 3 painful days of doing this. This creating 100,000 annotated images this way took a couple of hours. Right? And so the question is, now we have these annotated images. You have the image on the left is the original image. We have the image on the right is the segmented image, right? The mask that we say, where are all the plants? We use this to train a machine learning algorithm. The bottom line is, it works. Right, the details don't matter. I mean, that's an upcoming paper that you can look at. From our perspective, it works, right? And we could do a lot more. We can improve things in terms of distinguishing between. If you notice, this is actually an image when the machine learning algorithm essentially is going in and painting, if you will, pixel by pixel as blue what it thinks are the plants, right? You see that here, all the plants reasonably well are all the pixels are painted blue. We are still working on distinguishing between foreground and background because we only want to identify the front 6 plants that the camera has. So we want to distinguish between the front 6 plants and everything else. We also want to look at individual plants. This is called segmenting out, distinguishing between each individual plant. So keep watch. Zaki and Kaushik might have more results very soon that we're able to identify individual plants. Now, how am I doing on time? Okay, I have 1 minute left, so I'm going to wrap up quickly here. So we tried 3 different things, and I'm going to race through these 3 different things in the next minute or so. How far away can we get away from creating this synthetic data? First, we tried using different genotypes, and the genotypes that were there in the field. It works. Next, we tried using a digital plant. So this is James Schnabel's group created virtual plants on the computer using something called Plant Factory Explorer, and then we used those same things to embed them in a background and create these things. It worked too. And then we tried going to a different species. Again, this is instead of using. So the idea is that we don't let to waste any data that has been well annotated. And so James Schnabel's group had a very well annotated sorghum data set that we used. We did the same thing on using sorghum data set, and I just want to show that it actually works. And interestingly, this goes back perhaps to link to Erin's talk just before me. When you use sorghum plants as the domain adaptation, as the data set to train this, you're actually capturing the brace roots much better because of the fact that there are tillers in sorghum, right? And so this is something, again, something that I never realized, but when I gave the talk to the group, they figured out that this is the reason, right? So domain adaptation done carefully, done thoughtfully, is going to be very, very, very helpful. So there are other ways of doing this. In this case, domain adaptation is I can actually take someone off the street and tell them this is the stock, right? And they can actually mark the stock. But if I cannot, for some of the traits that we are interested in, you need an expert. And this is especially for plant pathology, right? If I want to figure out diseases, I can't just pick someone off the street and say, tell me what disease it is. You need to have an expert, right? And so how do we minimize annotation load of experts is something that Aarti Singh will talk about. So please tune in for our next week's talk. And with that, I'll conclude here, right? So there are several approaches available to the plant science community on minimizing our annotation load. I talked about one thing, domain adaptation. There are a few more called active learning and self-supervised learning that perhaps Aarti will talk about in next week. I want to conclude by saying that it's important that we, as the plant phenotyping community, create, curate, and share data sets for the community to grow. Right? It's important that if we put in the effort to create these data sets, it's important that we actually publicly share these data sets. With that, I thank you for the opportunity to talk, and I think Erin and I will be happy to take any questions now.
I have a question for Baskar. So do you think that your algorithms and the kind of domain adaptation approach on the whole plant would work on, I'm just going to use myself as an example, the base of the plant images that are, a single plant image? Because I noticed you're pretty far back. Is that important for your training, or is that just an artifact of how it was imaged?
Yeah, so it's a specific of the experiment. Okay. Because this is Pat Schnabel's large-scale experiment where they have either placed a stationary camera 6 feet or 8 feet away from every row. And the reason for placing it that way is that you have enough of an imaging window to see the complete plant grow from seedling all the way through all its stages without having to move the camera. and that's why it's far off, right? But I foresee that this can easily be translated, if you will, to focusing only on say the brace roots, right? So, and then if you are able to have indoor imaging of brace roots, and then also capture reasonably what the background ought to look like, an empty background, and transfer this, I'm reasonably certain that it'll work.
Nice, thank you. That's beautiful talk.
Okay, while we're waiting for more questions, I'm going to ask Erin a question right back.
Please do that.
So I really enjoyed your talk, especially as a mechanical engineer. I think the connections to me are pretty strong in terms of the mechanics of it and the stress strain curve that you have. When I teach my class, I teach that. So a question for you is the first part so second part of this, right? So when you talked about the root brace, sorry, the lodging it from the root, right? So not the green snap, but the other one. Does soil composition matter here?
So the short answer is no one's looked yet. I'm sure it does. I'm sure, that's one of the things that, and one day, once we get these devices a little bit better set, I would love to tag on to Genomes to Fields and actually start to get that information about how different environments are contributing to this. I mean, I can't imagine it doesn't play a role. It has to, right? But we haven't quantified any of that yet.
I think you alluded to the fact that civil engineers and geotechnical folks have some understanding of this, and I'd encourage that it might be fairly simple that you measure something about the soil, take a core sample, and then you can unravel the effect of that particular soil on that, right? So.
Yeah, absolutely. And in Delaware, we tend to have really sandy soils. And so, if I could go to our different kind of stations throughout the state, but it changes just our percent sand really. But yeah, absolutely. That's funny. Where I'm headed is learning about soil.
Awesome. Thanks.
Yeah. Hey, Erin, my name is Clayton. I had a question for you related to that stress strain curve that you're talking about. And I was thinking in terms of the actual brace roots themselves. You'd mentioned kind of that flexibility, that ability to bend was really critical to the standability. And I'd be curious if you all thought about or investigated taking an individual brace root and doing like a tensile strength test on it, like pulling it apart. Will that create a stress strain curve? And would that potentially correlate to the whole plant then as well? Because then you could just cut a couple brace roots and potentially see how the whole plant would react too.
Yes. So we have a paper on bioRxiv right now where we've taken the brace roots and we've done 3-point bending. Instead, so some of the issues with doing tensile tests is how you attach it.
Yeah.
Right. And
really juicy, really fast.
Right. And so we're still sorting through some of that data. It's interesting. When we take the structural mechanics, they vary across the whorls within a plant, within a genotype, and it's because of the differences in diameter. But then between genotypes, we do see differences in material properties. Whether those correlate, I'm trying to remember. We looked and it wasn't great. It didn't give us a great indication of the plant mechanics from the brace root mechanics.
Okay. Well, I'm curious too, just from a purely corn background, then were you noticing broader differences between, say, like stiff stalk versus non-stiff stalk populations in terms of those mechanics, or if that was even something you explored?
So we did actually, I'm gonna totally put Ashley on the spot. Ashley is here. She's my postdoc. She looked at some of these classifications, and Ashley, correct me if I'm wrong, but there was no relationship in stiff stalk and non-stiff stalk in terms of what we were getting out.
Correct. I think I believe you're correct on that.
Okay.
I missed the first part of his question, but then from what you just said, I believe you're correct.
Yeah. So we didn't see these nice like stiff stalks actually measured stiffer, or it was kind of interesting.
You're referring to the phylogeny.
Yeah.
And there was no clear groupings as far as lodging.
So I have a quick question for Baskar, and this relates to the Envirotron facility that's at Iowa State. Have you looked at whether you can do the same kinds of plant segmentation from like above or other angles, or everything you were showing that I saw there was mainly from the side of the plant? Have you looked at other angles?
Yeah, excellent question. I think this is also some of the questions that people asked at Kaushik's prelim exam a couple of days ago. And so, yeah, the method, the notion of domain adaptation, all it requires is you have synthetic data of a different but related domain, right? So, and that's so here the decision of how to figure out what the indoor images were made by the fact that our imaging protocol was from the site. However, you can actually use images from a different angle, from the top, especially if you're looking at phenotaxy and stuff like that. It should work, right? So we haven't tried it out, but I'm fairly certain it should work.
So I would think that and this is just sort of me thinking out loud here, but it would make sense to me that what you're taking the data from and what you're projecting it onto would need to be sort of at the same sort of angle. Is that
so
the basic problem?
That's right. So you need to train the machine learning algorithm to figure out patterns in the images that you actually care about, right?
Yes.
And so for it to learn the correct patterns, it has to see similar patterns in a synthetic data set, right? So some variation you can robustify, if you will, the model, and that's actually an active area of research. But it has to be something similar, right? However, there's always exception to the rule here. There are new methods that essentially do a sequence of domain adaptations. That is, you take a cat and dog classification and then say train to something slightly different, right? So canines versus non-canines. Then you move it to something else, animals versus plants, and then you train it, and you can do this sequence of domain adaptations and actually move very far away from what the original data set is.
Cool. Thank you.
I think. Can I ask a question too?
Please.
Yeah. So, in terms of your first part of the talk, right? So, does it matter where the force is applied along the height of the plant? Especially because if I understand correctly, the lower part is more mature and so has more cellulosic content, whereas as you go up, it's going to be less. And so, do you have to keep track of that? And so, if you do across genotypes that have different heights, then do you have to look at the internode, say at the node, say I look at the fourth node and push there?
Yeah, so first I'll say that all of this is based on Euler-Bernoulli beam theory, which basically we calculate the flexural rigidity, flexural stiffness, which takes into account the height of the load. So it accounts for that. What it doesn't account for, though, is your second point, which is the height of the plant. And one of the things that we've gone back and forth about is, do we test all the plants at the same height, or do we test the plants all at the same proportion of their height? And we elected to do the same height just because we don't want to move it up and down, from plant to plant. But you're exactly right. We don't really understand how the distribution changes based on plant height. We do know that height matters, right? Taller plants tend to be bigger and stiffer overall, but we're measuring them at the same height as a shorter plant. So it's a conundrum that we haven't quite figured out the best way to approach yet.
Awesome. So I might have some ideas. We can talk offline.
Fantastic.
Very good. Any connections made through these Phenomics Fridays? That's the whole point. So I hope we do this again, make more connections. Thanks for coming. Thanks to the people in the room and the people online for taking time for this. Please spread the word, send the flyer out to people, invite others to come, and thank you for being here today. Thank you especially to Baskar and Erin for presenting their seminars for us. Happy Friday.
Thanks.
Thanks, everyone.